mutant population with phenomics data. So today I'm going to use TRIM as the example. So first, I also just like PJ, I like to have acknowledgement, to the funding agent as well as collaborators. Totally there are 4 PIs, including 2 rice breeders, I myself, and Dr. Yue, who is a molecular biologist, and of course, many of the young scholars in these labs. As I mentioned yesterday, there are a lot of tech mutant population, but some of them, there are phenomics data. Here are all the numbers, and altogether, we still have less than 200,000 lines. So this is the website of our database. We may search by query sequence or keyword, including putative knockout genes or activated genes or mutant line number or phenotype, including simple trait or QTL trait, as well as segregation ratio. So here is the infrastructure of the whole mutant population. In molecular biology lab, generate lots of tDNA lines, and then in the TERI, GM field, breeders, collect seeds as well as do the phenotyping, and all the data goes to our database, and also we perform flanking sequence analysis, and then those data are stored in TRIM database. And then the seed are all stored in a very good resource center, for seed storage. And then all this data may be available for people to search, for either forward or reverse genetic analysis. So right now, there are all number, available right now in our database. So when you are using our database, please notice that in the first about 20,000 lines. We are using tag 4 vector, so they are promoter trapping as well as knockout function. But then from the line starting about 1800, then we have promoter trapping, knockout, and activation function. So we do have 8 copies of 35 base enhancer, which is very close to left border. So in our database, please notice where are the left border, because outside from left border, then we have 15 kb regions that may be activated. But then because the vector is very long, so if that is out from red dB, then only 5 kb region may be activated. That is why, although we do have data, upload to NCBI, but we still prefer, if you are using our database, if possible, go to our browser, because it's easier for you to interpret the data. So in the material we are using Taiwan 67, and first, the tDNA insertion low side, the average is less than 2, and also the top 17 almost do not jump. So here are the GM fields that we propagate all these seeds. And we have many of these mutant type, as you, phenotype mutation, as you may tell here. So about 18% of T1 population show obvious mutated trait, and about 15% of the screen plants show gas activity. Here are the variations, in QTL traits. That's the whole table, but I will tell you how to make the search. So here are the T1 mutant, because sometimes in the field you may find out for some mutant lines, they are dark green, they are severe dwarf, and the leaf are white. So some of them might be correlated. So we also perform the analysis, and we indeed found out some of them have very high correlation, which is indicated here. And because right now, in our TRIM database or in Japanese top 17 database or in Grameen or in Yue-ie, we do have different phenotype categories. So also in our database, we prepare a comparison table, a big Excel table, so that you may find out, okay, from the view of rice breeders, okay, like TO term, whatever number here may match in top 17 or in our trim database, what the definition are, so that we may cross-talk. So if you first please log in our database, you just have to sign your name and fax to us. We will give you the login information. So you may search either by locus, or line number here, or by blast. So if you go by blast, then you may use DNA as query, so BLASTN, or protein sequence, then T-BLASTN. You may also perform BLAST search using query sequence. Like here, because I work on Yue-ie gene, so I am using Yue-ie1 as the query sequence, so I BLAST. So please notice here, so you may guess that there are only one intron for this short gene, okay, 2 exon here. And because I just mentioned that outside from the left border, they may affect 15 kb, outside for right border only 5 kb. Okay, so wherever you see this arrow, okay, in the arrowhead it means right border, right, and the other side is left border. So come out from left border like here, then they may, okay, activate the nearby region for about 15 kb. So right now here we have 3 lines, okay, so for each of them it's 10 kb. So altogether this is 30 kb. So whatever gene you are interested in, okay, exactly there are in either side there will be at least 15 kb region, which you might be affected by this integration. So you can see here that for this one, okay, they may activate this gene, and then all this up to about here. For this one, about the same thing, and for this one, they may activate only about this region, and then from here to the other browser area. But anyway, you may see that in this 30 kb region, there are several integration sites here, and okay, so 6 trim lines in this 30 kb region, or 11 genes in this region may be activated, and 2 of them may be knocked out. Okay, so please be aware how to use our database. And also, while you perform the search, please use IE, the Internet Explorer. Okay, Mozilla or other browser cannot, somehow the code is different, so I'm sorry that we may only use IE. So if you are interested in ethylene, okay, so you can just type, okay, either putative knockout genes or activated genes. So you can also, like right now here, I make choice of both. So you can see either knockout K or A activated. Or you can just type root. You may also see a lot of them. Putative activated or knockout mutant, okay, are available in our database. As once again, we have about 100,000 mutant lines, and about half of them we already have Frankie sequence data. And of course, you may also perform phenolic search. Select here, okay, for QTL, the most obvious will be plant height, panicle number, and heading date. And so here, okay, by the categories, okay, we have about 11, okay, different category. And if you click on one, then another area will pop out. So like here, this is leaf morphology. Then we have, okay, a lot of description of the mutation phenotype. Or like you can also click on leaf color. So for this search, okay, I search for dark green leaf, white leaf, and short leaf. So you can see how many of them are listed here. And then with the number, then you may ask for the seeds. Or say like panicle number in our field, okay, single seed descent. So the average panicle, or okay, say effect panicle tiller number is 13. Okay, right now I type on for anything, it's more than 55 tillers. So you see we have up to now 22 of them. So see this one, we have almost 90 tillers. Okay, 90 tillers with very short panicles, and for most of them, okay, they are very short dwarf. Okay. If you are interested in heading date, in our field, because in Taiwan we may have 2 cropping seasons, so the average heading day is 70 to 90 days, depending on different cropping season. So if I type, okay, heading date is smaller than 50 days. So you can see, okay, 2 of them is only less than 1 month. Okay, so they will be very useful if, like Yano-san just so he came in. So if he's interested in very short photo period, I mean, not sensitive to photo period at all. So within 1 month they may head. Okay, so they are very interesting mutant lines. Or if I type on longer than 115, then we found out, okay, 4 months. Okay, they will not heading in our, because if the variety we are using are not sensitive to photo period, not sensitive to temperature. However, we still see those very prolonged, okay, heading day. So they are also very interesting. All the seeds are stored, okay, in the National Research Center. So they are short-term storage room or long-term storage room. So for if we put here, maybe we may store for at least 10 years. For here, well, maybe 80 years to 100 years, so very long. So this means that for all the T1, okay, for the seeds coming from T0 plants, we always store them in long-term storage, and then because we propagate them, so part of the T1 seeds we will store in short term, so ready for distribution. For distribution, it's not work in my lab, because in my lab we are only responsible for bioinformatics data. So if you are going to ask for any seeds, okay, please go to this website, okay, tdna.bts.asia.edu.tw. Okay, so this is a website, and so you still have to log in here, okay, and then so you may perform search, or you may ask, okay, apply for seeds, okay. So they will respond to you by email. Okay, if they send seeds to you, usually they will send 30 seeds, T2 seeds only, no T1 seed, because they are treasured. We have only very few, because it's only coming from the the single seed coming from tissue culture. So only 30 seeds, and also we will remove the parent lemma. Okay, so next. Embryo, okay, I mean endosperm and embryo, so that if you are ready, please apply when you are ready, so that once you receive the seeds, okay, please germinate them within 2 months, because we already removed the outside, so that it's easier to go through the quarantine. Okay, because for rice, it's also, I mean, very important maybe for your nation's agriculture. So I'd like to introduce our GM field. Okay, so this is our GM field. Okay, as you may see here, okay, it's a necked house, big necked house, and once it is 1 month after transplantation, we grow 3 by 4, so altogether 12 plants. So the breeders will work in the field. Wherever they see mutant, say like here, okay, so they will make a mark. So this means that if you see the white paper here, it means in that location, the rice breeder already performed the screening, and they found out mutant phenotype already. So we harvest by single seed descent, and here are all the breeders, including the very senior one, and some other juniors rice breeders as well as workers. It's really a big team, so that we may provide very good phenomics data, and also we propagate the seeds and then ready for distribution. Thank you very much.
Thank you, Yue-ie. such a large collection of resources for functional genomics. So, if there's any question from the audience?
Usually, if you may find out the phenotype record, this means it's already been propagated. Okay, we already have T2 plant. So this means maybe the seed are almost ready or already ready. But because in my side, I'm only responsible for the trim and I'm not responsible for the seed, so I dare not really tell you, but I'm also one of the PI. So for me, it takes me about 2 months to apply. Okay.
What are the what are the quarantine requirements for requesting seeds from Taiwan? Is there any? Do we need to send some certificate for importation?
Yeah,
seed
I
importation?
think for this TTRSC, they acquire MTA. For my side, we don't need MTA, but for this means that if you ask for seeds, you have to sign. First, sign NTAs. Second, if any researchers in USA, it's almost impossible to send out. I mean, we send out, then they was returned back. So because quarantine in each country are different, I think Genome also have similar problem. It's easy to send to some country, but it's very difficult to send to some country. Yeah, so please bear with us. Yeah.
Okay. Okay, thank you very much.
Okay, thank you.
And with this.